This will be the 6th lecture in our lecture series for this semester, and it will cover extraction. This will be a pretty short lecture. It's a fairly basic process with some very simple mathematics, and it shouldn't be too difficult compared to distillation, and it will really just kind of add more to our separation repertoire. We'll start with discussing the different kinds of extraction. We'll talk about what the partition coefficient is. We'll talk about what back extraction means, and we'll talk about drying agents. An extraction is basically a separation of mixtures that can be liquid and solid in nature. Usually it's solids dissolved in the liquid, but really it can be a partition between any two phases of any of the different kinds of states that you have. So it could be liquid solid, gas liquid, liquid liquid. Usually you don't see gas solid, but I'm not saying it wouldn't exist. It's just that it's not as practical. But most commonly it's going to be two immiscible liquids, especially in our type of work. Partition is based on relative solubility within two solvents, and we'll talk about what we mean by partition and partition coefficient in a moment. Techniques include both macroscale and microscale. With macroscale, you use a separatory funnel, which is the large pear-shaped sort of glass structure with a stopcock on it in your drawer that has two different openings. And then you have the microscale, which is basically just taking centrifuge tubes and Pasteur pipettes and using different kinds of setups with that to do what you want to do with it, and a picture will demonstrate better. So with macroscale techniques, the mixture is usually dissolved in solvent 1 and then placed in a separatory funnel. Then you have an immiscible solvent 2 that's added, and then you stopper it with one of those Teflon plugs, or it could be a glass one, depending on the type of glassware you're using. And then you agitate it. You kind of shake it up, like I'm trying to think of a good example of what that would be like. I suppose the best example would be like those crafts fair projects that you do with small kids, where they put different beans and stuff in a bottle and you try to mix them up, and so you're just kind of, shaking it up all around in there and trying to get it thoroughly mixed. It's the same kind of idea here, except that every once in a while, and what's not shown on this slide, but that we'll talk about before we go down and do it, is that you need to tilt the separatory funnel up with the tip, with the stopcock on it, facing upward at an angle, and you have to open the vent to vent the gases. This prevents it from becoming unsafe. And there's an example of this in the safety manual from the ACS that I posted for additional notes on lab safety under that content item on iLearn. As far as what happens afterward, over time the two mixtures separate, but in one of the layers there will be some of the item that was dissolved in the organic solvent that transferred into the other solvent based on its solubilities. And so over time you can drain off those solvents and separate the materials. With the microscale technique, you use the centrifuge tubes, and it's the similar idea in the sense that you can add your dissolved materials in the solvent and add the immiscible solvent, and then you can put a cork in the top, usually not rubber stoppers. Rubber stoppers leach into organic solvents in our lab, so if you can avoid using the rubber stoppers, you're probably going to have a lot better results with your materials. You're better to use a cork because they tend to put less material in there. And then again, once it's shaken up, you can vent that at an angle and by removing the cork, and then you can let it settle, and you can use a Pasteur pipette to retrieve the materials from the bottom. Usually the more dense layer is removed. We don't remove from the top. You remove from the bottom by sticking the long Pasteur pipettes, the 9-inch, down into the bottom and drawing it up from the bottom, and then you transfer it into another tube. And you can do several of these extractions like this. It works really well when you have a small amount of material, and this is often the case. So to calculate the partition coefficient, which governs how much goes into solution on in either case, between the two solvents, how much will be extracted when you do the extraction depends on your partition coefficient. So the partition coefficient is equal to the quotient of the solubility of compound A in solvent 1 divided by the solubility of compound A in solvent 2. And for most typical purposes, solvent 1 will be organic and solvent 2 will be some kind of an aqueous material or some non-organic solvent. It's usually an aqueous solvent of some kind in our lab anyway. Now, because of these things, you can actually calculate it in a little bit different way, because oftentimes what you'll have is you'll have a solubility, and solubility can be written as mass in unit volume instead of like a molar concentration. So it works better for you if you work with it in that regard. Excuse me. And so you will basically use this proportion here to calculate that. And it comes from basically taking a literal version of what's here above and working it out. But this is all you really have to understand here, is that the product of having the expression of the quotient of the mass of compound A that's present in solvent 1 divided by the mass of compound A in solvent 2 multiplied by the quotient of the volume of solvent 2 divided by the volume of solvent 1 becomes equal to the expression that is equal to the KD. So you can work all the problems by basically saying that the KD is this little expression right here. And that's pretty much how you work all these problems. I've actually left some problems on the note page for this particular slide for you to go through and work, and you can check your work with Dr. Krause's slides that he puts up on iLearn in the pre-lab materials for everyone across all sections. So that's what I would encourage you to do. I think instead of me working all of them out, I think you just need to work them out and then check your work, and you'll make sure that you're getting it that way. Because if I work it all out, then you may not try enough, and I can't come up with enough examples before the quiz. So anyway, that's what I would suggest you do. As far as, how we go about it to get the aqueous, usually if you've got some solid in there, it's probably some kind of a carboxylic acid or some kind of an amine or something like that. And so what's useful in those cases is to use a reaction solvent. And so we'll pick something like HCl or ammonia or sodium hydroxide, or it just depends on what you're trying to do exactly, but any of those things could really work. We typically use strong things though, like strong bases and strong acids, so it'll usually be like HCl in our lab or sodium hydroxide, and it's usually it's not usually super concentrated either. It doesn't necessarily have to be. You kind of want a gradual sort of change so you can get multiple extractions out of it. It just depends on the situation though. It depends on the specific experiment. But these general equations remind you how the acids and bases are traded off, and it shows you basically the process going on with what's happening at the functional group in question. And all of this will be decided by the pKa. So that's an important thing to keep up with when you're talking about extraction stuff, is looking at your pKa's and seeing which will happen first. As far as a process for this, it's pretty simple. You have your organic acids or bases or a neutral. They're dissolved in a solvent. You extract with an acid, in this case, has been selected, and you're going to get some aqueous phase here with either your additional charge from having a proton added, or it could be, in some cases, simply just turning it into a salt, essentially. Because oftentimes what's happening is you don't have any that's been deprotonated yet. Particularly like with benzoic acid, you're trying to get the proton off of the carboxylic acid group. So you add, by adding the sodium bicarb that we add in that experiment, you're actually turning it into a water-soluble salt, and it's going into the aqueous phase. And the other items that don't have that group, that aren't as readily protonated by a weak kind of mixture like that, sodium carbonate, 10% by weight or something, sort of diluted like that, they're not going to travel into the aqueous phases easily. So that's kind of the principle behind doing this. And again, you pick the solvent system that works for what you're trying to get out. So it depends on the particular experiment. In back extraction, that idea is basically kind of always done. I don't want to make it sound like this is something special. This is all part of extraction. And what you'll find is that usually you're doing a combination of these things. I mean, it depends on what kind of results you're getting, but if you have, excess organic solvent coming out in your aqueous material, generally you try to save that and put it back and try to back extract so that you can be sure you've gotten it all. And oftentimes you test the pH of the material to see if you're still getting neutral sort of pH out, because if it's got the bicarb, it should be turning kind of a more basic color if it's bicarb heavy. So sometimes we check with litmus paper and things like that and see if we're close. And you try to kind of calculate based off partition coefficients and things to see if you're kind of around where you should be, solvent wise, so you're not wasting too much material. You can get into some little bit of calculation like that to see if you're kind of in the ballpark range of where you need to be. But it really does depend on the exact measurements for your solutions you're using. So if you're just even a 0.5 here or there off, it could really affect your outcome when you're trying to make a theoretical calculation like that. Anyway, that's kind of the idea. So you just keep doing it until you're satisfied that you have your stuff, and then you can kind of have your two separate solutions to go with from there. Now, as far as the organic phase is concerned, the organic phase that remains generally has to be dried. So once you put some water into it, there's a good chance you'll have a little bit of water in your solvent, because remember, everything has a solubility. And even though water and organic phases don't normally dissolve in one another, they do dissolve just a tiny bit. And that also changes based on temperature and other things. So you have to take that into account. What oftentimes I see people do, and I will say this is one of the areas you can make a mistake, is that they do not get enough water out of their solvent to begin with. You should always be looking at the solvent that you have in front of you, and you should see if there are some water droplets in it. And if you can see them, if you're If your solvent is less dense than water, it's going to be above the water. So water's going to sink. If you see water in the bottom in that particular circumstance, you have got too much water in there. You don't need to put a drying agent in it yet, and because that's too much water most of the time. Now, if it's like a little bitty drop, and I'm talking like it's just a speck, then that's different. But most of the time, you need to try to get that out with a pipette if you can, either by angling the flask or putting it in a centrifuge tube or something. And you need to try to get that out, because if you don't do that, then you will have a mess, because it gets really hard to put enough salt in there to dry all that out, and you'll be doing it for a long time. And even then, it may not be perfect. So that's something to keep, that is something to keep in mind and to think about if you want to be sure you're not going to have to use too much salt, because some of these aren't as nice to play with as others. They can be very messy in some cases. They can make interesting things on the bottom in terms of like sludges and stuff like that if you have to use too much. So it's always good practice to dry as much as you can before you have to do the drying agent. And as far as, stoichiometry goes, sometimes I do like to ask you to calculate the minimum amount you might have to use for something. And these kind of give you a general idea of how you can generate different sorts of inorganic hydrates from the salts. So you may just kind of want to keep these in mind. And probably if I do ask one, I'll give you this equation, but you will still have to know how to determine, by telling, from looking at it, you'll have to be able to figure out about how much you have to use. And sometimes I don't like those questions, but sometimes I do, and I'm not sure what kind of quiz you'll get on this material yet. So I'm just throwing that out there so you kind of remember to look over it. Now, I have always tried to find a good picture to show people, because they always come up to me with their flask. And this is one of the better ones that I've found, and I'm going to give my thanks out to UC Boulder for putting this up on their on their organic lab website, where they were drying out solvents in their labs. And this shows you kind of the process. So this would be for like mag sulfate or sodium sulfate. Those tend to look more kind of like these powders. The calcium chloride pellets are more spherical, but they still do the same behaviors. And so you can learn by looking at this kind of what to look for. So here, you see a lot of this clumping, and it's not able to move around. That's got water in it. Over here, it's still clumping, and you have some small dispersed clumps. That's still got too much water in it. But over here, if I can swirl it around and I have lots of free floating salt in there, that's dry. And that means we're done with the drying. So you want to look for this nice free floating crystals in the solvent of the inorganic salts. That's exactly what you want to see if it's dry. And if it's not, it'll clump up like that over here or over here. And it tends to look kind of milky, sludgy like this. You just don't want a whole flask full of that. That's why I'm saying, dry as much as you can before you put in the salts, because you can only get so much out at one time. Okay, well, that's pretty much it. We'll see you Monday and answer any questions that you have about extraction. It's a pretty simple thing.